Okay. Hello, thanks for the introduction. Today I'm going to be presenting on making good use of genomic, spatial, and exposure risk data in infectious disease transmission models, and I'll specifically be focusing on a method that uses multi-level pairwise regression models. So I'll start off with a little bit of background about how infectious disease epidemiology is traditionally conducted, and then I'll go into how genome sequencing can inform outbreak investigations, and then the sort of state of models that incorporate genomic and epidemiological data to study infectious disease transmission and quantify that in sort of endemic settings. I'll then go into a couple examples of my own work doing this sort of analyses. The first example will be looking at Klebsiella pneumonia in a long-term care facility, and the second will be looking at community-associated MRSA infections in a community. So to start off with, infectious disease transmission happens in complex systems. You have biological, human, behavioral, and environmental factors that can affect infection and transmission of infectious diseases. You can see a lot of different things that can affect that up on the screen right now. But today I'll be focusing basically on using the genetics of pathogens in order to study different risk factors of transmission within and between hosts. So transmission risks can happen on multiple different levels. You can have individual characteristics such as like sex, age, that make you either more or less likely to either contract or pass on an infection. And then you can also have pairwise or dyadic risk factors, which I think are talked about less often in infectious disease epidemiology, which are basically shared characteristics of someone who gives and gets an infectious disease. So it could be like living in the same house, being of similar ages, things like that. And then you have these sort of contextual or systemic risk factors, which are the characteristic of a time or place where an infectious disease is transmitted. So that could be like the age structure of a population. It could be a specific infection is just, there's an outbreak of an infection at a specific time or place, and that will increase each individual's risk of getting infected. So now I'm going to walk through how we identify different risk factors for transmission in traditional epidemiology. So basically, what usually happens is we recognize that there's an outbreak of some sort of infectious disease, and we define that basically as just more cases than expected in a given time and place. And then we examine the characteristics of who's infected and who's not. And based on that, we determine the risk factors for infection. And this can be done in a number of different study designs, such as case control, cohort, or ecological studies. And now just to walk through like a very simple example of an infectious disease outbreak. So let's say that your department hosts a potluck with 100 people, and a few days later, 40 of them come down with a gastrointestinal illness. So you bring in an epidemiologist, and she surveys everyone who was there and asks what they ate and what they didn't eat, and finds some concerning results with the potato salad, which is a very classic example of foodborne outbreak risk. She finds that basically of the 44 people who ate this potato salad, 38 got sick. So the risk of infection and exposed is 0.86. And then only 2 of those who didn't eat potato salad got sick. So the risk of infection of those unexposed is about 4%. And then you can calculate a risk ratio and find that eating potato salad at this potluck increased your risk of getting sick by 23.9 times. And this is relatively simple to do because we have a contained system. It's a single event, and there's a single source of infection. So you can use the traditional methods for this sort of outbreak investigation. However, transmission risks aren't always so easy to identify, and this can be for a number of reasons. You can have endemic settings where the disease is spreading, constantly throughout the system. And so you're not gonna have this like one single transmission risk. You can have many importation events. So this could even be like within a household. Say like a kid comes down with the flu and then their parent comes down with the flu. You might think that the kid transmitted it to the parent, but who knows, maybe the parent got it at work. And you can't really tell that from traditional epi investigation. You can have concurrent outbreaks with multiple different things causing the different outbreaks, open systems, people coming in and out. Some infectious diseases present with asymptomatic or persistent colonization, where someone's basically colonized or infected with an infectious disease but presents no symptoms. And you can have high prevalence of disease. So like 99 out of 100 people at the potluck got sick, it would be a lot harder to identify the source. So now I'm going to talk about how whole genome sequencing of the pathogens can help in these sorts of situations, inform infectious disease epidemiology studies. So basically, over the last 2 decades, the price of whole genome sequencing has dropped like exponentially on the exponential scale. As a result of that, the number of bacterial genomes being sequenced has increased exponentially. And now that we have the data, we kind of have to come up with these robust methods in order to incorporate these data into our studies. So a simple example of how we can use whole genome sequencing in infectious disease epidemiology is to identify outbreaks, right? So here's an example. It's a special care baby unit, which I think is like the equivalent of a NICU, but this study was done in the UK. And they have, so each line on this graph, so the x-axis is time, and each line is a single case of methicillin-resistant Staphylococcus aureus, or MRSA, which is a bacterial infection. And you can see that over time, they have a case or two, but then after about 150 days, they see 3 cases in 2 days, and they think, maybe this is an outbreak, maybe something is going on. Let's look into it. So they know all these cases match antibiograms, which is just like they're susceptible or resistant to the same antibiotics. So they might be genetically linked, but they don't really know. So they whole genome sequence all of these isolates, as well as other MRSA cases in the SCBU during this time period. And they find that indeed they are all genetically connected. And they find 2 more cases that they hadn't previously identified are part of this outbreak. And it's not just those 3 cases, it's all 15 of these infants. What they do next is they sample the parents and caregivers for MRSA. And they find that a number of those individuals have genetically linked isolates as well. And they're most closely genetically linked to the infants that they're caring for. So they've now increased the size of the outbreak by using whole genome sequencing, or they have increased what they can define as part of the outbreak that was already happening. They then sample healthcare workers to see if anyone is in the healthcare system is spreading this disease between the infants, and they find a single healthcare worker who has a genetically linked isolate. And so basically what they did here was more of a descriptive study, but they were able to identify a potential infection source of an outbreak in this setting by using whole genome sequencing. But what happens if you have a system that looks like this? So this is much more than 14 cases. This is a long-term care facility. And these are all carbapenem-resistant Klebsiella pneumonia colonization or infections. You can see sort of, it's small, but there's a lot of different importation events and a lot of acquisition events. And it's probably not just one thing driving these infections, right? So this is a data set that I will get into more later, but just like keep this in the back of your mind, how are we going to use whole genome sequencing to be able to like identify transmission risks in this more complex system and quantify those risks as well? So now I'll go over a few examples of published methods that integrate genomic and epi data to infer transmission risk factors in these sorts of endemic settings. So I'll go over 3 examples. I'll go through some of the limitations, and then I'll go through the 4th method, which I'll go into in more depth. So one thing you can do is compare those infected with a certain sequence type to those uninfected, or those with a different sequence type. So this is just, again, defining who is in an outbreak, and then you can more easily compare those who are not. You can also compare those who have a known direct transmission event to those without known direct transmission. And also you can like integrate whole genome sequencing into these sort of compartmental, probabilistic, susceptible, infected, recovered transmission models, which are common in disease modeling. And then finally, you can use pairwise regression models. So here's an example of a study that compared those who were infected with certain sequence types to those who were not. So basically what they did, this is like the entire ecosystem of healthcare associated infections in a hospital over a given period of time. And you can see there's a lot of different organisms. And then they were able to define, they whole genome sequenced all of these organisms and were able to define these clusters that you can see are dots connected with bars. And they did that by, basically they define them as if they had fewer, less than 2 or less single nucleotide variants separating each of the isolates. And a single nucleotide variant is just like a single mutation that occurred at some point during the transmission cycle. And then once they defined the clusters, they used data mining of the electronic healthcare record system, as well as machine learning to identify potential infection sources for each individual outbreak. So for example, they found that an outbreak of vancomycin-resistant enterococcus was associated with interventional radiology and the injection of sterile contrast. They found a Pseudomonas aeruginosa outbreak was associated with gastroscopy, and they found multiple outbreaks of different organisms associated with being in the intensive care unit. So they're basically able to identify risk factors for each individual outbreak in this example. Here's another example of that second sort of method that I described, where they look at characteristics of those with versus without evidence of direct transmission within a facility. This time they're looking at Clostridium difficile, which is another bacterial infection common in healthcare systems. And they basically, again, defined a direct transmission link as less than 2 single nucleotide variants between a potential donor and a recipient of this infection. And they found that things like older age, a longer time in the facility, and then certain ribotypes, which are like genetic types of C. difficile, were associated with being more likely to be a donor or recipient of direct transmission within the facility. And these are all individual level factors. Finally, this is an example of a study that incorporates whole genome sequence data into susceptible, infected, recovered model, transmission model. And what they were doing here is they wanted to determine the contribution of various transmission routes of C. difficile in a healthcare system, whether that be known contact within a ward, known contact within the hospital, or like background contamination of the ward, hospital, or community infections. And by utilizing the whole genome sequencing data, they were able to determine by strain type what was the most common routes of transmission of C. difficile in this hospital. So for example, like sequence type 1 was more likely to be from a known donor, while the rest were more likely to be from hospital background. So these are all super cool, but there are some limitations. First of all, a lot of them require SNV cutoffs to infer transmission, which is relatively simple to do, but can result in a false positive and false negative identification of transmission events, and requires a lot of assumptions to be made about the transmission ecosystem and the pathogen. A lot of them require data on uninfected controls, which again is relatively simple to do in a contained healthcare setting, but it's going to be much more difficult once you get into the community setting, and using traditional public health data sets usually only contain data on cases of infectious diseases. Some of them, like the SIR model, require many assumptions to be made about transmission dynamics. And I think in that paper, they did a good job of that, but it's like a lot of work to figure that out for each different pathogen. Some of them, like the machine learning example, are computationally expensive and sort of black boxy. You don't really know what's going on in the models. A lot of them don't necessarily account for the effect of super spreaders on model outcomes, which I'll talk about more in a second. And none of these models are able to identify individual dyadic and system level risk factors within an entire like infectious disease ecosystem. They usually focus on just one of these. It's also, again, when we're looking at community spread, going to be hard to incorporate spatial data into these models. So now I present the pairwise regression model, where instead of on the individual level, we're looking at, or comparing those infected versus uninfected. We're taking everyone who's infected and comparing basically everyone to everyone else to see how closely they are infected and see what sort of factors are associated with closer related genetic distances, basically. So yeah, in this model, every observation is a pair of infected cases, and the outcome is how closely related the isolates are. And you can define that in a few different ways, which I'll go through. And then you can use individual dyadic and contextual risk factors to predict genetic relatedness, which we're kind of thinking of as a proxy of transmission. So in this model, the outcome is the log SNP distance between two individual isolates. And then we can incorporate dyadic risk factors, such as how far apart they live from each other, or like the time difference between the infections. And we can include individual risk factors such as sex, age, occupation, education. And then we also are going to be clustering by space and individual with these spatially correlated random effects. Because each outcome is a pair, you're going to have individuals represented in your model multiple times. So you need to be able to control for that. Again, this is like the example where you might have a super spreader who infects 100 different people. They're going to be very closely related to a lot of people in the data set. And so we don't want their characteristics to be like over accounted for in the model outcomes. And it can actually like artificially narrow confidence intervals if you don't have these sorts of things. And it also controls for unobserved heterogeneity on a spatial scale, which is important in these kinds of models. So this model was developed by Josh Warren at Yale, and he has an R package available on GitHub for incorporating these sorts of models into your own research. And this is an example of sort of a proof of concept, a paper by Josh Warren, where he used this sort of models to look at tuberculosis cases in the Republic of Moldova. So they're all whole genome sequenced. The outcome of the model is the log patristic distance, which is another measure of how closely related two isolates are. And then he uses these pairwise covariates, such as distance between village, age difference, and then individual covariates, sex, residence, working status, and education, to see if any of these things are related to how closely related to tuberculosis isolates are, and if there's evidence of transmission risk factors. And unfortunately, in this specific model, none of the covariates are significantly associated with patristic distance, but it was a really good proof of concept of how to use these models. So now I'll transition over to some examples from my own research where I use these sorts of models. The first one is using individual contextual and dyadic information to identify infection sources for carbapenem-resistant Klebsiella pneumonia cases in a long-term care facility. So let's break that down. Carbapenem-resistant Klebsiella pneumonia, or I'll refer to it as CRKP, is a highly antibiotic-resistant healthcare-associated infection. There are high rates of morbidity and mortality in long-term care facilities where we have a lot of people with a lot of underlying conditions who are very they're more likely to get these sorts of opportunistic infections. And but there is more to learn about how this bacteria is transmitted within these facilities. So the data set I used surveillance testing for CRKP of all patients within a single long-term acute care hospital over 1 year, and they were tested for CRKP on admission and every 2 weeks. Since this is one of those pathogens that can cause asymptomatic colonization, we need to be serially testing them to see if they have acquired the infection. So this was done from June 2012 to June 2013. It included a total of 260 patients throughout the year and 15,000 patient days. Every CRKP isolate that was isolated was whole genome sequenced. And it turns out that 98% of the individuals in this study were colonized or infected with CRKP at some point during the study period. So we have this very high prevalence ecosystem. So the objectives of my study were to develop statistical models that utilize whole genome sequencing data to determine infection risk factors and quantify them. And I wanted to identify risk factors on the individual pairwise and contextual level to determine infection risks of CRKP. And now we're coming back to this nasty looking data set. It's actually a nice figure. Not done by me, sorry, that was a previous student did this. So what I did is I developed this pairwise regression model, and I used 3 different outcomes representing genomic relatedness for the outcome of these models, which are supposed to be a proxy of direct transmission. And the next slide, I will get into more about how these are defined. But there's single nucleotide variant distance, closest genomic distance, and membership in a genomic cluster. Then the covariates I used were shared time on the same room or floor of the facility, and time difference between infections, which we can think of as dyadic covariates. I looked at antibiotic use of the potential donor, which would be an individual covariate, and then time period in the study, which we can think of as a contextual covariate. And to be able to visualize more what's happening in these models, here's an example of 5 individuals in a facility A, B, C, D, and E. They are all infected with CRKP kind of sequentially in this order. Whole genome sequencing cluster analysis shows us that A, B, and C are clustered into one group and D and E are clustered into another. And then this is what our outcome data is going to look like. So basically to walk through it, patient A is not going to be a recipient in this model because they were the first one to be infected. They probably came into the facility with an infection, and so we can't tell who gave it to them. But everyone else, we can make some sorts of inferences. So for patient B, the only person infected before them was patient A, so they're going to be their closest potential donor. We know they're in the same cluster, and the whole genome sequencing data tells us that their isolates are 4 single nucleotide variants apart. Then for C, they could be infected by either A or B, but because they're more closely related to B, they're going to be their closest donor match. And then, but they are in the same model for, the same cluster for both A and B. For patient D, could have been infected by A, B, or C. They're not actually in the same cluster as any of them, but who knows, maybe some like interhost, like evolution was happening, or maybe we missed a transmission link, but we're going to say that because it has its closest in SNP distance to A, that they're their closest potential donor. And then for E, they could have been infected by anyone, but D is both the closest in SNP distance and the only one in the same cluster, so we're going to call them their closest potential donor. So these 3 columns are basically going to be the outcome in 3 separate models. The SNP distance model will be a log linear regression, and the closest donor model and the same cluster model will be logistic regression since they're all 0s and 1s. And each of these models will include a random intercept for the potential donor. So the results of this study showed that shared space and time was associated with genetic relatedness in this facility. We were able to interpret the outcomes of each model kind of similarly. And they were all consistent in showing these patterns. So what they showed is that room sharing during this pairwise exposure period, which is the time between the last time the first patient tested negative and the first time the second patient tested positive, if they shared a room during this time, they were more likely to be in the same cluster, they're more likely to be the closest potential donor, and they had shorter SNP distances than other pairs in the data set. Which makes a lot of sense. There is previous studies that show having a roommate who is infected with CRKP increases your risk of getting the infection. So that was kind of check. We also showed that overlapping on the high acuity unit during the same exposure period was also significantly associated with closer genomic distance and cluster co-membership, where this was not necessarily true for other floors of the healthcare facility. And this also makes sense because there is previous work showing that being on a ventilator or having higher needs can also increase your risk for different sorts of healthcare associated infections, because basically you have more ports of entry, but also more hands-on work needed by healthcare workers who are thought of to be vectors of these sorts of transmissions. Also, in this particular healthcare facility, they weren't able to incorporate as many infection prevention control measures in this specific unit as they were in other units. So again, this all makes sense, and it's pretty cool that we can pick it up with these models. We also showed that less time between positive tests was associated with closer genomic distance. So in conclusion, infection control and prevention efforts in this hospital and other ones should be focused on roommates of CRKP infected or colonized patients, as well as residents of high acuity wards. Additionally, we showed that pairwise regression models can integrate individual dyadic and contextual epidemiological data to be used to quantify infection risk factors in an endemic and complex transmission setting. So now I'm going to transition on to another example of work that I'm doing, which is still in progress. I'm not going to have results for you right now, but I'd love to talk to you about the methods. And so this is kind of taking these methods to the next level into a community setting where we don't have as much information about uninfected controls. We don't have, serial sampling, and we have this spatially heterogeneous study population. It's an open system, and So what we'll be studying is community-associated methicillin-resistant Staphylococcus aureus, which is referred to as MRSA, which was once mainly an infection of concern in healthcare systems in the United States, but since the late 1990s, it's been spreading into community settings in the US and around the world. It can cause a variety of infections, such as skin and soft tissue infections, which is the most common in the community, but can also cause more severe infections, such as bloodstream infections, pneumonia, and sepsis. It is estimated that 10,000 deaths a year in the United States are caused by MRSA infections. And MRSA can also, like CRKP, asymptomatically colonize people. And it's estimated that 1 to 2% of people in the US are asymptomatically colonized. The antibiotic resistance of MRSA makes it more complex to treat than susceptible Staphylococcus aureus, and it's becoming more and more antibiotic resistant over time. So we really want to find ways to identify risk factors for transmission so that we can prevent these sorts of infections from happening in the first place. There are many known risk factors for community-associated MRSA infections, including at the individual level, such as participation in sports, IV drug use, recent incarceration, homelessness, and HIV infection. We also know on the community level that household crowding, living in an urban area, poverty rates, and prevalence of formerly incarcerated individuals in a neighborhood are also associated with community-associated MRSA rates. However, we don't really know much about pairwise risk factors or exactly how these infections are moving through communities and how to quantify which risk factors are causing more MRSA community infections. So I hope to address this by using this data set that we have from the Cook County Health System, which is a safety net healthcare system in Cook County, Illinois, which is the county that includes Chicago and its surrounding suburbs and includes about 5 million residents. So we have data on all MRSA infections that presented and were diagnosed at this healthcare system, which includes 2 hospitals and many outpatient facilities from 2009 to 2022. So in total, we have 1,300 community-associated MRSA cases out of that. And we have demographic and risk factor data for each of these cases, including drug use, homelessness, incarceration history, age, sex, race, ethnicity, and census tract. And each of these isolates were also whole genome sequenced. We also include community data in our analyses that come from the Census and American Community Survey on neighborhood characteristics. And we also have information on incarceration from the Illinois Department of Corrections and the Cook County Jail on a zip code level. So based on initial analyses, it's clear that there are disparities in community-associated MRSA rates in our study area. You can see here this graph This graph shows community-associated MRSA rates in 2009. And this one shows proportion of households living below the poverty level from 2007 to 2011. And you can see that higher rates of MRSA are associated with higher rates of poverty. And it's also associated with many other things, but here's just an example. So the objectives of my study will be to identify the transmission drivers of community-associated MRSA in Cook County and describe how MRSA is moving through the community to create these spatial patterns and inequities that we've observed. So I will be using, again, a pairwise model that evaluates how factors such as shared risk factors, geographic distance, and time between infections influence the genetic similarity of isolates from community-associated MRSA infections in hopes of revealing potential community transmission drivers. And my hypotheses are that shared risk factors, such as both individuals having experienced homelessness or both having used illicit drugs, as well as geographic proximity, may be associated with more closely related MRSA isolates. And I hope to be able to quantify how each of these are affecting the transmission system. And then hopefully with these outcomes, we can help inform public health interventions in the community and inform whether they should be more risk factor based or neighborhood level programs to prevent infections in the future. So to wrap everything up, my conclusions are that whole genome sequencing is more accessible than ever and has a great potential in infectious disease epidemiology. However, there is a lack of robust methods that quantitatively integrate this genomic data with epidemiological and spatial data in order to identify and quantify drivers of infectious diseases, especially in endemic settings. However, I'm hopeful that these sorts of pairwise regression models can identify dyadic, individual, and contextual risk factors of transmission for multiple pathogens in multiple settings. Just like to take a second to acknowledge and thank my advisor, John Zellner, and the rest of his lab, as well as Evan Snitkin, who is a professor in the Department of Microbiology and Immunology and a collaborator on all of these projects, as well as the rest of my dissertation committee and our collaborators at Rush and Cook County Health, and Josh Warren at Yale, who's developed a lot of these models. They're my sources, and I'm happy to take any questions. My emails are here.
If you're online, go ahead and put them in the Q&A box, and if you're in the room, feel free to ask.
Yeah.
I guess I didn't follow it close enough. With your cohort, it include asymptomatic patients and or the test for the SNVs automatically include people that are
For the CRKP data set it included asymptomatic carriers. Yeah, I also did a sensitivity analysis that only included people with invasive infections, as that's what's normally caught in these facilities. And some of the models were able to pick up some of the risk factors, but others were less able to pick up the identified risk factors. So it also shows the importance of this sort of broad sampling to get the most informed results.
So if they included asymptomatic factors, what was your definition of an infected?
It's basically anyone who was, I think it's it was the infected, the clinical infections were part of a different data set. So everyone was being swabbed for asymptomatic, for colonization every 2 weeks. But we also had information on, and that was part of a scientific study, not necessarily informing clinical decisions, but then there are people who, had a UTI or pneumonia or something. And so the healthcare workers tested them for what was causing that. And that was considered a clinical infection if they presented with symptoms and then were diagnosed with CRKP. And that was kind of separate. Does that make sense?
So, I'm John. one thing too, that in these systems that I think we kind of struggle with is that the difference between colonization and infection is very delivery, right? It's not like latency, like if you think of COVID or tuberculosis, where there's this period before you get sick. With colonization, we kind of assume you're infectious when you're colonized, but you're infected. It's almost like an epiphenomenon of colonization, but there's nothing necessarily about infection and just being more infectious. And so, you know, this kind of selection that happens on invasive infections can be selected on the risk factors for getting an invasive infection, which can obviously be, you know, problematic for getting a transmission.
So for the Mercer Cook County study, for the health, for the non-Mercer control samples, how will you pick up those samples? I don't think you can include all down.
We don't have any controls in
You
that
don't
study.
have, okay.
And that's why this method is kind of cool, because we only need cases in order to,
Okay.
so we're not like testing or like looking at who's infected and who's not and what risk factors like make you become infected. It's like basically taking two cases and like assuming, and you have to like interpret it all under the like guys that like both people are infected. What makes them more likely to be closely connected or not? Are they in the same like transmission like cycle or not? Yeah.
And for all the models you mentioned that we need to measure SMV distance. What kind of metric are you using for defining SMV distance?
Yeah.
And, have you tried other methods so that you can test the robustness of it?
Yeah. So it's basically just the number of variants that different within the core genome.
Just
Just the core genome. But yeah, it's like there's I think a lot of opportunity to look at different outcomes and see.
Yeah.
if they
So you don't use like I heard that some people are using like a Jacob, metric, if you convert to 201, that kind of because we have a bunch of for each chromosome, because you are using whole genome, right? So for each chromosome we have
it's the pathogen.
yeah, and then we can convert to a 01, convert to like binary vector, and then because if it's binary vector, we can use a Jacob, a metric to define the distance. I don't know whether you have tried that, or not.
So yeah, so you're sort of saying like something like a Hamming distance, right? Where you have
Something like that, yeah.
two sequences and you have these, you count, instead of we're summing up the number of same distances,
Yeah.
but you essentially say these are the ones that are different.
Compare with the
Like shared variants.
yeah,
or-
compare with the reference genome.
Right,
Because SMV event network is rare,
yeah.
right?
So Hannah, we moved quickly past this just for the interest of time, but Hannah did look at 3 different outcomes, right? And so one was like more algorithmic clustering, which I think kind of gets closer to that. And that method turned out to be kind of the best of the 3 in terms of, telling us what's, about these risk factors, because as you might imagine, it's like the SNP, if you look at a histogram of those SNP distances, it's kind of bimodal, right? Where there's like a bunch that are really close and then everybody else is kind of far. And there's not a lot of information in the far ones. They're just not related. And so, yeah, the way you choose your clustering or your relatedness ends up mattering a tremendous amount.
So, and also for the pairwise model, again, it's linear model, right? Have you ever been considering non-linear, dynamic effects?
Yeah, we did a log linear model. So basically
But
like
still it's not linear, right?
Well, there are these more kind of involved models that get at some of the evolution or even just a drift, but they require a kind of density of sampling that in those data sets just doesn't exist, and also like really strong assumptions about the molecular clock. So the idea here is to kind of go in a more conservative direction and try and identify risk factors using these data. But obviously it's kind of subject to kind of limitations, but where those data are available, that's a great approach. But like particularly say for this community MRSA study, those data just are never going to come along, and you have MRSA everywhere, right? And the transmission is not a very well-defined event because it's colonization, it's kind of more like a cloud, right? And so we can't get at that. And so these methods are really good in those situations. Whereas if you're doing like HIV in a small population where everybody's exposures, then those more kind of phylogenetic models become very, very useful.
I also think there's like kind of a beauty in the simplicity of the SNP distance model, where we were like talking with Josh Warren, who developed these, and he was like, everyone just wants to use SNP distances. And I think it's like important to show if that is an okay thing to use or not, because it's like relatively easy to get at, and like people who are not in like large research institutions and stuff can do these sorts of models to like look at these community infections in their own communities and things like that. So
and also for MRSA data, the data you got is just cross-sectional or patient panel data to track the progression of each individual, which way? Do you have longitudinal?
No, there are a few patients in the data set who have more than one infection, so we can look at those. But mostly we just have information about the person at their time of infection.
Okay.
Yeah.
Question about like variables personal and project. So there's with that for hypothesis we could like illicit drugs and like homelessness. I was wondering with that kind of ask the introduce bias when you're trying to discover the model, because it's considered.
What kind of bias do you think? Because you think there's like highly associated or
Like unwillingness to just.
oh, yeah. Yeah. So the data we have on that is from people's healthcare records. So I am sure there's a lot of underreporting. So yeah, we'll have to like take that into mind when interpreting the results. But that's a good point, yeah.